Okay, in this video, we're going to go through all of the physical ways to control microorganisms. One of the first ones that we mention most because it's so common, immediately I'm going to cook it. I'm going to kill it with heat. That's what we've been doing for a long, long time, is that we're going to kill it with heat. So there's two types of ways to control microorganisms with heat. There is dry heat, flame, fire. Bunsen burner, which we don't use. We use the incinerator, which is a bit safer than the open flame of the Bunsen burner, but both are a form of dry heat. When you are heating these things up, every microorganism is going to have a different thermal death point. So if you have this microorganism exposed to heat at 10 minutes, what's the lowest temperature that's going to actually kill it off in 10 minutes? Is it 10 minutes at, I don't know, 40 degrees Celsius? Is it 10 minutes at 70 degrees Celsius? What's the lowest temp that it will take to kill off that organism in 10 minutes? And then the other is the thermal death time, and that's the length of time needed at a given temperature. So if we're going to set it at 70 degrees Celsius, I have no idea if that's hot enough or not. I'm just making these things up. But if we set it at a certain temperature, whatever, 90 degrees Celsius, how long does it take for that time, for that death to occur? How much time does it take at a set temp? So they are kind of flip-flops of the other. Both ways, we're looking at killing the organism. One of them has a set temperature and the other one has a set time, and you're looking for the temperature. So again, this is dry heat. And then there is moist heat. And so that's what our autoclave is going to use. It's not only going to use a wet heat, it's going to use steam, but it's also going to use pressure. And the combo is a 1-2 punch to knock stuff out. Autoclave is the most popular thing to use to sterilize stuff, to kill the vegetative cells, to kill their endospores, to kill all the microorganisms. The autoclave is the way to go. That's what we put our surgical instruments in. Dental hygiene tools, all that stuff goes through the autoclave and sterilizes it. And it's going to use steam and pressure. And there are two different ways. I'm not quite sure if I put them in here. They mention them in the textbook. I'm not going to get that specific on the test. But they do mention two different ones that the autoclave can do. One is gravity, where the steam comes in at the top, and then the steam is heavier than the air, so it displaces the air goes out the bottom. And then the other one is a vacuum style, where it sucks the air out, and then it adds the steam in afterwards and pressurizes it. Both of these, some autoclaves can do both of them. I think it said most autoclaves can do both of these different types of cycles. One cycle is better to get everything, wrapped items. The vacuum method is a little bit better. So if you have a package of all of your dental hygiene tools, all the things in there, and I'm not dental hygiene, so I don't know the names of all the fancy tools, but all the names of the little tools, and you had them packaged up, in your little kit, and you wrap it in the kit, and you put your autoclave tape on there, and you put it all in there. That other method with the vacuum sealing, where we suck out all the air first and then steam and pressure, that will penetrate packages better than the other gravity displacement method. So different methods for different things. Indicators. I was telling y'all about the autoclave tape. So this is what we use in our lab. So you can see here, before, it looks like just regular old masking tape. And then after it's gone through the autoclave, you see the hash marks, the black lines that indicate that it got to the proper temperature, and this is sterile right now until you open up the package and expose it, open up the top and expose it. But at the time, that tape indicates that it has been through and it's okay. There are other ways to indicate that things are sterile from the autoclave. So the textbook mentioned a spore test that's going to have a strip or a suspension of endospores, and it's Geobacillus, Germophophilus. And if those things get killed or not, if they're still growing, your autoclave's not doing the job that it should be. And then another third method is the diac tube, where it's going to have a pellet, and if your autoclave's doing what it's supposed to, it should melt that pellet inside the tube. So these are two backup ways that you could check every so often just to make sure that it really is doing exactly what it's supposed to be doing in the autoclave. Pasteurization. We've talked about Pasteur, I feel like every other month. We're bringing up Pasteur every other chapter. So pasteurization is when we are going to reduce the load of bacteria inside that substance, whether it is milk, maybe it's pasteurized orange juice. In this example, we have pasteurized milk. And I want to say, was it 2 chapters ago? Our lady, and she was pregnant, and I forgot her name, but she was pregnant and she was trying to do all the things right, and she got the organic food and the locally sourced milk that wasn't pasteurized, and then she gave herself listeria. If she had gotten pasteurized milk, then she would not have gotten sick with the listeria because it would have knocked down the bacteria loads and killed them off. So there are 2 different types. We have high temperature, HT, high temperature, short time. So we have 72 degrees Celsius, so a high temperature, and that's going to kill off, and here we are, it's going to list all the things that it's going to kill. Campylobacter, this guy, Listeria, E. coli, H1, O157H7, the bad one, TB, all these things, Salmonella, all sorts of bacteria that when we pasteurize it and we heat it up to these high temperatures, it's going to kill these off. They're not completely gone though. This stuff isn't sterile. They're just brought down. So we heat it at 72 degrees for 15 seconds. This is high temp, HTST. Then there is the ultra high temp, UHT, where instead of 72 degrees, we're going up to 138 degrees, and we're going for an even shorter time. Instead of 15 seconds, oh no, sorry, I lied. We're doing 2 seconds longer, 2 seconds longer. So we're going extra high heat, and we're going for two more seconds. So 17 seconds total at this extra high heat. These are good. I have kids, I got 3 kids. It's a busy time here at my house. My kids love these packaged milks. And so these are done, just this exact brand, by the way, Organic Valley's prepackaged milk. If you've ever gotten those pouched kind of things and they're pasteurized like this, this is the ultra high temp, and these will last in situations that may or may not have refrigeration. So these will last longer. If you've ever checked the dates on the stuff that you get on the fridge section versus this stuff, you are going to have a longer date on this one. Speaking of my children, I hope you cannot hear that fighting in the background of this video. Yay! All right, we're gonna move on to refrigeration and freezing. I was telling y'all, right, if you had some bad meat, if you had some meat that had some bacteria in it, and you put it in the fridge or you put it in the freezer, and then you brought it back out of that it's in stasis when it's in the fridge. It's in stasis when it's in the freezer. If it's a certain types of bacteria are susceptible to the freezing. They get exposed to the freezing and they'll die, but some of our pathogenic ones, they're not. They're just in stasis and they're just hanging out. And then later, when you bring them out of the freezer, when you bring them out of the fridge, they warm up and that bacteria grows. So refrigeration and freezing are ways to keep things in stasis, to just put them on hold. If you're going to thaw stuff out, it is recommended that you not do my method. Don't stick it on the counter. Don't do that. It tells you officially to put it in your fridge. That's the safest way to defrost your food. That's not what happens at my house because everybody's hungry and we want our food yesterday. So I try and go the fastest way possible. But for safety's sake, put your food out of the freezer and stick it in the fridge and de-thaw it that way. Take the time, do it right. But we know what the right thing is supposed to do. Yeah, ultra low freezer, and put things on dries, keep them really cold. If you are doing a research study on something and you're freezing your samples, that's the way to go. Let's keep going. Pressure is another thing. We've talked about heat, we've talked about cold. What about pressure? Pressure we mentioned in the autoclave. Pressure is going to kill stuff off. So our atmospheric pressure is 0.1. 0.1 when you're at sea level. So if you, the beaches are open in Florida actually today. The governor decided to open the beaches tonight. on, today is April 17th. So the beaches are open, and if you decide to go down to the beaches at sea level, the pressure in the atmosphere is 0.1. If we crank up that pressure, and now we're at 100 millipascals, that is way more than sea level pressure, and that is enough to kill most vegetative cells. But as we said, the endospores, those sneaky little buggers, they may survive. But this is a way to, if you've got an active infection, hyperbaric oxygen chamber might be a way to combat some of those, because what happens in those, not only are you under pressure, but you're breathing pure oxygen. So you can be in the chamber, or maybe you just got the tube, but it's giving you pure oxygen. And at that level, and at the higher atmospheres, with that pure oxygen, what it's going to do is it's going to saturate the tissues. And these tissues that may have gotten poor circulation, they may have gotten hypoxic because of the infection and inflammation go into those tissues. We talked about inflammation with our diabetic patient and his foot, and he was getting inflammation in his foot, and then eventually just went numb and the tissue went necrotic. This hyperbaric oxygen would force the oxygen to those tissues and hopefully combat the infection that takes place there, because it's going to increase your white blood cells, your neutrophils and macrophages. It's also going to form those free radicals that happen when you deal with oxygen, like peroxide and stuff like that. Those things are toxic to a lot of substances, and not every organism can handle those kind of free radicals with oxygen. So a hyperbaric oxygen chamber is a way to possibly combat deep tissue infections and kill those things. I can't remember exactly what we were talking about besides gas gangrene, if they mentioned anything else. No, I don't see anything else in the textbook besides Clostridium perfringens. Gangrene is a way that you can combat that. There are risks. So oxygen toxicity and the pressure may affect delicate tissues like your ear or the lungs. So there are risks. It's not a fail-safe perfect thing, but it's a way to, another method to combat things. Speaking of pressure, the textbook has a story about a lady that came in. They don't give us her name. Oh, excuse me. She's in Ohio, and she comes in the hospital, and she's got some serious symptoms. She comes in with blurred double vision. She can't swallow. Her eyelids are starting to droop. She's got abdominal cramping, nausea, paralysis, dry mouth, weakness of the facial muscles, difficulty speaking and breathing. The doctors immediately know with all these symptoms that she probably has a case of botulism. She's probably got toxin from botulism. So they called up the CDC, had the CDC overnight them some anti-botulism medication, and more people come in. And it turns out that they were all exposed to some potato salad that was made with canned potatoes in a home canning kit. And they weren't used with a professional pressure canner. They were used the way that a lot of us have just been told to use, is to boil the water, and you'll put your items in the can. Like, you wash your cans, you put them in the boiling water, you put your materials in the can, and then it's so hot it's supposed to kill off everything, but it's not gonna kill off the endospores when you do this. And as the substance in the can cools, the pressure is going to drop because everything condenses and gets smaller as it cools down, and it closes the lid on your can. That's basically how canning works in a nutshell. But that method, again, it didn't kill off the endospores. So later, when they used this canned good and they just opened it up, made the potato salad with it, and they didn't cook it after they opened it up out of the can, those the botulism toxin was in there, and it got everybody sick. And I want to say the original lady passed away, but the other people that had ingested it, I think they made it. So pressurization is another physical way to kill the pathogens. Another way has been something that we've been doing since forever, throughout the ages, and that is desiccation. And there are lots of different ways to do it. You can just set things out in the sun. Literally just set your grapes out in the sun and you're going to end up with raisins. If you have, I don't know, like $30, I think is what I spent on mine. You can get a food dehydrator and it will have adjustable little temperature settings and a hot, fan that blows over your food and it will dry out the food. If you take out the water, none of these organisms can live. You have to have water unless they're in an endospore form, like we've talked about before, and then later they'll come back. But desiccation, drying these things out, if you remove the water, you're killing the microbes. You might not kill their endospores. And when conditions get changed, maybe you accidentally have moisture creep back into the container that you had of, I don't know, dried fruit, your raisins. Maybe you didn't close your mason jar all the way and the lid got open because your kid decided to raid the mason jar and didn't put the lid back on there. And so the moisture and the air got in the jar. Now the microbes can grow again because it's not desiccated anymore. So stuff can get back in there if they get wet, but the whole purpose of dehydrating is to pull the water out of the cells and dehydrate them. So again, you could use the sun, dry things out in the sun. You can use a food dehydrator. You can use a freeze dryer, which is really cool. They have these home freeze dryer units. They are super duper expensive, like over $2,000 expensive for these things. There are some preppers out there that do have these that, I mean, they're amazingly cool. What they do is they freeze the food, they flash freeze it. And when they do that, it's going to use not only the cold temperature, but it's also going to desiccate this out. The water that's in there is going to go out as vapor that it removes out of the substance. So it's freezing it and it's dehydrating it at the same time with the freeze dryer. It preserves the food slightly better. The process of using a food dehydrator, the food is exposed to the warmer temps and it takes a while, hours, sometimes days to dehydrate certain things. And so you lose a bit of your vitamins when things are warmed up for a long time like that. So your food is a little bit better preserved with the freeze dryer. If you happen to have one of those, that's awesome, and I'm jelly. But both of these are both methods to desiccate, dehydrate the food. And then these last guys are going to use, we learned about isotonic and we learned about hypertonic and we learned about hypotonic. And hypertonic, the stuff outside the cell was a higher tonicity than what's inside the cell. So that's what we're using for all of these methods. If you like beef jerky, I love beef jerky. Beef jerky, everything, it's salted meat, it's salted, covering the meat with the salt will pull the water out. It is hypertonic and the salt is going to draw the water out of the cells and they're not going to make it. So we've been preserving food with salt for ages. And then you can also use sugar as a way to preserve food. Both of those, salt, sugar, are also additional ways to use hypertonic situations to pull the water out of the cells and kill them, desiccate them. Radiation comes in two forms. Both of them we have learned already will mess with the DNA of a substance and it can cause mutations. So there are two types that specifically they're talking about for us to sterilize things, to make things safer for us. This first one is ionizing radiation. The CDC, the FDA has signed off on this. This is really big in Europe. I don't think it's really taken off so much here in the US, but it's using gamma rays to irradiate the food. And so the way it works, the food is going on this conveyor belt and it passes through the machine and it irradiates the food, but the food is going to be okay. It's going to be in between there, in between the two sheets, so it's still safe. If you see this symbol, this is the symbol for this, for gamma radiated food. It's, again, super popular in Europe, and if you see that, that's what that means. Non-ionizing radiation. This is one of the tests that we can do in our lab at the very end because we can use UV. So UV is a non-ionizing type of radiation. We know that it can also cause mutations like these thymine dimers. And where the thymines are bonding to themselves instead of the adenine across the way. With this can cause mutations and this can sterilize stuff. We have a UV lamp inside of our cabinet, just like is pictured here. And that will, everything that the light touches, like Lion King, is not gonna make it with the UV. So there's UV in our cabinets, or I call it the fume hood. There's UV in the UV light in there. Sometimes they have UV light filters for water. I've seen them. I've never used one. Myself, personally, I use a membrane filter for my water, but there are, I can't remember the name of it, something pen, like Steri pen or something like that, where it's supposed to be like a straw or something, and it's got a UV light, and it claims to sterilize the water as it passes past the UV light. But I really want to test in the lab and see if that really works. I would like to figure that out. But UV radiation is a type of non-ionizing radiation, and it can be used to sterilize surfaces. Sonic waves. If anybody has that type of toothbrush, a Sonicare toothbrush is using this. So high frequency sound waves are going to lead to cavitation, and it's going to cause air bubbles in these things. So formation of bubbles inside the cell. And of course, that's not going to be good for the cell to have air bubbles inside of it. So it's going to disrupt the structures and kill off the cells. So Sonicare toothbrushes, a sonicator, like in this picture, can cause that. So that's one more thing that we can use to get rid of the microbes. We have already mentioned this guy before, the HEPA filter. We mentioned it on the biosafety level 4, that you have to use HEPA filters. We have these in our fume hood, in the safety cabinet. We have HEPA filters. And so they get stuff down to 0.3 microns is their effective pore size. And so it will get rid of all of these other things. It should get rid of bacteria. It should get rid of the microbes, their endospores, and viruses, teeny tiny viruses. So these are HEPA filters. Here's the safety cabinet. Again, we just saw one here with the UV light. This one's closer looking to the one that we have in our lab, so I picked this picture. But this is the safety cabinet. You can see all the different BSL levels, and what it's going to do is control the air. It's going to control the air so it's not flowing out into your lab. It's going to suck it up into the fume hood, and make sure that it's not going to, if you're handling something that could be easily aerosolized and you're trying to transfer it and stuff, you don't want that blowing back in your face, right? So you want that fume hood, that cabinet, to suck the air away and filter that air. So that's what these guys do with their safety cabinets. And then membrane filters are going to be our last stop. These come in a variety of sizes. We mentioned that the HEPA filters were 0.3. Some of our membrane filters are going to be 0.2. They can go smaller. Our normal bacteria is 1, like 1.01 is the average size of the bacteria. So if you are at a membrane filter of 0.2, you're filtering out bacteria shouldn't be able to get through there. And you could go even smaller, again, because viruses are smaller than bacteria. When we were mentioning about the pore filters and trying to get down to just the viruses when we talk about virus chapter, you could use a membrane filter and do that too. You might want to filter stuff if you didn't want to heat it up and kill the organisms, if you still wanted the solution that they're in. Or maybe you're going to filter out a larger bacteria and you want to keep the smaller bacteria, or you want to filter out the bacteria, but you want to keep the viruses. Whatever reason that you want to use this, you can use a membrane filter to do that. These tables are great to have all of the information in one place. I'm not going to read it to you because we just went through all of this stuff, but all the different ways using fire or cold, pressure, drying it out, radiation. They don't have, there it is. I was like, where's sonication and filtration? Sonication and filtration. That's all the stuff for physical methods of control.